the second day of Ento24, and it is my genuine pleasure to introduce our second plenary speaker of the conference, Professor Steve Torr. Now, Steve joined LSTM in March of 2013, and prior to that, according to his LinkedIn profile, spent 26 years and 7 months at the University of Greenwich, where he was studying the ecology of tsetse flies, the ecology and behaviour of tsetse flies for improved control of both human and African trypanosomiasis, which you may otherwise known as sleeping sickness. Now, Steve has worked with partners, throughout his career, has worked with partners throughout Africa and the UK. And last year, his group was awarded the prestigious Queen's Anniversary Prize for the group's research. So we are in the presence of Tsetse royalty. Now, I won't take any more of Steve's thunder, but before I hand over to Steve, what you should know is one of three things. So when you see Steve wistfully looking off into the distance in his own headspace, you can be rest assured that he's thinking of one of three things. Number one, tsetse flies. Number two, fishing. And number three, Manchester City Football Club. Now, while we would love to spend the next 45 minutes listening to Steve's latest catch or his thoughts on what happens to Manchester City once Pep Guardiola finally leaves, this is an entomology conference. So we will welcome Steve to the floor and listen to his presentation on tsetse fly ecology and control. And please, a round of applause, please.
Well, thanks, Chris, for the kind introduction. And I'll resist the temptation to talk about Manchester City because I'm in Liverpool. But we'll talk about that later. So thank you very much for the NSOC for inviting me to the meeting and for everyone here this morning to come and hear a little bit about what I'm going to talk about. So I was mouse. There we are. Thank you. And a special thanks to Amy for all the lead up to this as well. So the title was Tsetse Problem, Harnessing Vector Behaviour and Ecology to Provide Solutions. And when I was first thinking about this, I was thinking about framing it as a question. Have we solved the Tsetse problem? And we wanted something short in the sort of adverts and so on. And that seemed a bit obscure. What's the Tsetse problem? What's he even going to talk about? So we ended up with this, but I'm still sort of framing it as a question. As you would expect, I'll talk about some of the progress we've made and how we're making progress by understanding Tsetse behavior and biology. But have we solved the problem of the research questions, at least in their practical aims? Have they no longer apply? So that's the sort of the thread of this morning's talk. So what's the problem with Tsetse? Well, of course, as entomologists, insects can do no wrong, and Tsetse, there's no problem with them at all. It's just the company that they keep. And their company is trypanosomes. And these are single-cell protozoa, small, sort of almost like snake-like protozoa when you see them in blood, and they cause sleeping sickness in humans. So the cycle is tsetse flies bite an infected host, either an animal or a human. It develops in the fly, infects another animal or human. And there have been two massive epidemics in the last century, one in the mid-40s and one in the late 90s. And the figures there, WHO figures, so something like 55,000 peak recorded cases in the mid-40s, and the most recent one in the late 90s, something like 37,000, I think is the maximum per year. But that is in very, very difficult settings. That last epidemic was in an area going from what is now South Sudan, Uganda, DRC, Angola. So very difficult settings, very turbulent times for those countries. And so all the cases that are reported, they're probably missing something like 90%. And those 90% will all have died. So this figure here showing something like 35,000 a year having sleeping sickness in the 1990s actually represents something like 400,000 people dying of this disease. So in these epidemics, it really is a very, very serious problem. So where is the problem? So in fact, there's not one problem, there's three problems, which we'll talk about. So this line across Africa, which roughly follows the Rift Valley divide between East and Central and Southern Africa. So to the west of it is a form of sleeping sickness called Gambian sleeping sickness. It's called by T. brucei gambiense. It's a chronic disease. From infection to death takes something like 2 to 3 years. And it's an anthropanosis. So tsetse flies are biting infected humans and then biting another human and spreading the disease accordingly. The other striking thing, again, in the general sort of chat, we would say it's a disease only affecting sub-Saharan Africa. But in fact, it only affects really severely relatively few countries. So in the case of Gambian sleeping sickness, over 2 out of 3 cases actually are in the DRC. Other countries are Angola, Uganda, South Sudan, Central African Republic. Those carry the main burden. 95% of all cases have just been in those 5 countries. Then to the east and south of that line, so these are countries like Tanzania, Zimbabwe, Uganda. We have the acute zoonotic form of the disease. So the trypanosome here is TB rhodesiense, circulating naturally in wild megafauna, so buffalo, elephant, bushbuck, and it's an acute disease in humans. So the disease progresses from infection to death in a matter of weeks. Again, so the country that has the main burden of this is Uganda. Something like, again, over half of all cases have been in Uganda. And also Uganda is the only country actually has both forms of the disease. So the yellow blocks in both of those blocks are in fact Uganda. So Uganda and DRC are much of the focus of this disease. And then just as a sort of background, it's different from, say, malaria or some of the other diseases, vector-borne diseases you might think about. There is no vaccine for this. There's no prophylactic drugs, and it's fatal if untreated. So the only way of dealing with it is if you get it, there's a very difficult and complex treatment, 2-week IV course for most of the time. 5% of the people die of some of the drugs that are administered. And unless you receive those treatments, it's fatal. And then the third part of the problem is that tsetse flies also transmit a range of trypanosomes which infect livestock. So right across sub-Saharan Africa. So all of these are places we've worked over the years: Ethiopia, Kenya, Uganda. These are all cattle in tsetse-infested areas. These cattle are at risk and do, in fact, get nagana. And in some ways, this is a bigger problem, those much less high profile, especially in the School of Tropical Medicine. So again, it's very difficult to know how many animals are getting it, but the sort of figures that you'll see in the literature are something like 2 million cattle killed every year by it, 35 million cases, over 2 billion. So a really, really serious disease for livestock. So in the same way that there are 2 diseases of the two human diseases separated geographically, there are vectors also associated with those 2 forms. So Gambian sleeping sickness in the west and central parts of Africa are largely vectored by riverine Tsetse. And this is a scene from Masimanimba in the DRC. The actual name of the place refers to sleeping sickness. This is one of the areas we're currently working in. And this is the sort of typical habitat and areas of infection. People coming down to the river, collecting water, washing, fishing, and Tsetse are in the habitat, the vegetation along the sides, and bite humans. And then in East and Southern Africa, so these are the vectors largely associated with Rhodesian sleeping sickness and also with nagana, the animal disease. So these are associated with savanna habitats. These are species like Placina morsitans and Pelidipes, and they typically sort of associated with the large, naturally with these large parks like the Serengeti, Tarangire, Mana pools, for instance. So that's a sort of a little bit of a background, and that's the problem that we are talking about. And the problem was first identified and linked to actual Liverpool itself. So John Everett Dutton, who was born in Bebbington, which is a small town just on the other side of the Mersey, opposite Liverpool. Was born there, grew up on the Wirral, and was actually educated as a clinician at the University of Liverpool. And then he, at the same time, it's Liverpool, LSTM, Liverpool School of Tropical Medicine, is celebrating its 125th birthday. Seems to be a birthday that goes on for 2 years, though, I've noticed. Nice. Anyway, so in the very early stages, expeditions were going from Liverpool to the tropics, and one of these Dutton was on, went to the Gambia, and he was the first person to describe seeing what turned out to be trypanosomes. It was on the Gambia River, hence the name Gambiense. So he called it, originally was Trypanosoma gambiense, but he had also recognized the earlier work of Bruce, who had described Trypanosoma brucei in animals, in livestock in South Africa. So the school and scientists at the school have been working on this for over 100 years. So again, sort of thinking about that 100 years, lots of the literature is, of course, framed about the problem. So these are some of the books on my bookshelf that I had sort of inspired some of the titles. So there we have The Tsetse Problem in Northern Nigeria. This is Ford's book, the long title then, A Study of the Tsetse Fly Problem. Lots of the early literature frames tsetse flies as a problem. Africa's bane. So it's not very enthusiastic. But then there's another lot of literature that is much more like the natural history, entomologists thinking about it, and obviously rather attracted to it. So natural history of tsetse flies, distribution of abundance of tsetse, again, sort of echoes of that early ecology literature. So what attracts entomologists to tsetse flies, including me? So first of all, it's got some very odd biology and physiology. So for a start, it doesn't lay eggs, and we don't have free living larvae. All the larval stages are actually developed inside the mother, and she lays this massive L3 larva, which is actually bigger than her, paradoxically. So it's like a human giving birth to an 18-year-old. So weird. They only do this once every sort of roughly 10 days, so it's a very, very slow reproductive rate. Very odd biology. And then associated with that, they only feed, both the males and the females only take blood. So they're not pollinators, they don't take sugar from plants. So again, following from that, there's no carbohydrate source to their source for energy. So again, their underlying physiology is very different. So sort of in the nerdy entomologist way, oh, that's interesting. Then perhaps less professionally, but very true, is the places where you go to study tsetse flies are very nice places to work. So these are some of the places we're currently working in: Bwaza Marsh in Malawi, northern Malawi, the Okavango Delta in Botswana, Serengeti, Murchison Falls. Now, who wouldn't like to go and study tsetse flies and insects in those sorts of places? And then the last thing, I think, in comparison to some of the other creatures and insects that we all work on, is they lend themselves to study. So for a start, in comparison to people working on anopheles and malaria that have to work during the night, then analyze their catches and process everything during the day, tsetse flies only active during the day, very short period of activity. In effect, we basically have a working day in the field, if we're doing work with tsetse flies, from about 3 o'clock till 6 o'clock. So leisurely breakfast, chat to your mates, go and catch some flies, go and have a beer. Again, that's sort of slightly related to their biology. We also have some things which I think are, again, fairly unique for some of the vectors and sort of applied aspects of entomology. For instance, we have the ovarian cycle of tsetse flies means that we can probably age them to an accuracy of about 48 hours, right up to 40 days of age. So all sorts of things about age structure, population structure, we can do. And we've known how to do that for something like 50 years. We also, again, it's a large insect, so a lot easier to dissect and look for infections, especially in the pre-molecular age. And again, the sources of energy and sort of doing energetic studies. We know where all the energy and all the resources come from. Those are blood meals. And we can look at how that is processed, and that's all converted to fat. So you can do some really neat sort of physiological studies on wild insects. And the last one is they are big, robust insects. So for instance, if you're doing behavioral studies, you can follow them, you can track them on this hoof of an animal here. You can see a tsetse fly in the foreground, Stomoxis just behind it. There's a host it's feeding, and there's all sort of interactions between other vectors, other biting insects, and the hosts. And because they're nice and big, you can put little spots of oil paint on them, multiple. You can do really elegant, large-scale, multiple mark release for capture exercises. So again, the literature is full of this sort of stuff. So I think that attracts us to tsetse flies as well. So that's the problem. We have some nice tools. People are interested in working on it. Is it a difficult problem to solve? So one way you can think about this is the R0 number. So I think all of you will remember R0 was a big part of our lives a few years ago during the COVID pandemic, where we were all we were regularly briefed on what the R0 number was for COVID. So those that don't know, the R0 is basically how transmissible a disease is, how many people are infected by one infected person. So at the time of the COVID pandemic, very early stages, the R0 number was about 3. So one person infected about 3 others before we had all the interventions. So how does that compare to sleeping sickness and other diseases? So first of all, just give you a sense of some of the other vector-borne diseases. So malaria has a very high R0, very variable, but in the 10s. Dengue down to about 3, so something close to COVID. And Tsetse and trypanosomiasis is really close to 1. In fact, in this particular paper, it seems that it shouldn't, Gambian sleeping sickness shouldn't even exist. But of course, this is a range around this. So some places the R0 is above 1, and it persists as we observe. So on the face of it, the disease should be relatively easy to tackle. What about controlling the vector itself? Again, going back to that peculiar biology, the life cycle is very long. They're just producing one larva every sort of 10 days. That larva burrows into the ground, pupates. That takes about 30 days. So every 40 days, a single fly produces another one. And then, of course, every 10 days after that. But it's a very slow process. Contrast that with mosquitoes. So Culex, egg raft of hundreds. Of course, there's some attrition and losses to all those larvae and pupal stages, but nonetheless, a very fast cycle and lots of. So again, it should be easier to control Tsetse than, say, mosquitoes. And then putting this into a framework, John Hargrove, publishing in Medical and Veterinary Entomology. Excellent journal, highly recommended. There's a framework, and this sort of gives you a sense of how hard you have to hit a Tsetse population to eliminate it. So the naught there is basically the population will not, if it's naught, it's not growing. This is all on a log scale, so 1 there, 10-fold increase in a year, 2, 100-fold increase in a year, and so on. And no matter how fast the pupal rate is, what we can see is that if you kill more than 4% of the population, Tsetse flies cannot persist. And that 4% includes all the natural background mortality that applies to a Tsetse population, to its natural predators, to natural diseases. So basically, you only have to give this population a bit of a nudge and it should collapse. So it looks like the Tsetse population problem is solvable. So 2012, there was a London Declaration on Neglected Tropical Diseases, and this is where we were in 2012. And 2 of those aims at that time, this was by governments, donors such as Bill and Melinda Gates Foundation, disease endemic countries, all came together and said, By 2020, we want to get down to less than 2,000 cases reported a year. There were other metrics, but that was one of the simple headline figures they wanted. So that's the blue line there. And they also wanted to eliminate it by 2030. So that is a really ambitious goal, that we don't want any cases anywhere. So that's the course we've been on. And this, if you remember, DRC is the country with most of those cases. And you can see these are the DRC cases here, and they pretty much mirror the sort of general pattern in the global cases. That's because that's where the problem largely exists. And all of this progress down to these low numbers that you can see in the DRC are the national program in the DRC working in very difficult places, screening literally millions. You can see on the left-hand y-axis, the values are millions. So that's something like 2 million people screened a year, and they're driving those down. So that whole program and that whole decline that you see here is not done with the help of any entomology at all. This is all clinicians, technicians going out, finding people, treating them. So Cat Rott's group at Warwick and been looking at various, and others been looking at, if we keep doing this, will we achieve those goals that we've set ourselves? And this is a recent paper. And basically, the red areas are places where we're not going to hit those targets. The blue areas where they're going to hit those 2030 goals. And this is if we keep on going at 2 million, roughly 2 million people a year, keep on doing the same old, will we get there? And the answer is no, we won't. But if we add vector control to that, if we start killing tsetse flies, which they haven't done up to that point, then we can achieve all of those goals. And that's sort of been the agenda for controlling and solving the problem. So about that time that the big epidemic was beginning to spread in the 1980s and 1990s, the new technologies for controlling tsetse were emerging. And the very first real sort of game-changing was work by Glyn Vale, who at that time was a PhD student. And he did this as part of his PhD. He looked at putting animals in pits, exhausting the smell, and see how many tsetse flies are tracked. And basically what he found was that the more animals you put in that pit, didn't matter what animal it was, it could be a ton of sheep, a ton of cows, a ton of goats, a ton of wild animals, put them in a pit, exhaust the smell, and you increase the catch of tsetse. And the more biomass that you have in that pit, the more flies you catch. Slight differences with males and females, species, and so forth. But turning that figure into a catch index, so this is expressing everything as what happens if you have no odor. You can see that for Pallidipes females, for instance, about 20 oxen catches about 20 times as many tsetse flies as having no odor. So that sort of, if we knew what that odor was, we could attract and kill tsetse flies. So that led to a whole series of research, largely in partnership with University of Greenwich, in particular the chemical ecology group, David Hall, catching ox odor on various resins, analyzing it by GC-EAG, and again, long story, very, very short, resulted in these chemicals. These, out of the hundreds, are important for, this is what tsetse flies use to locate the host. And if we release those at high concentrations, we can simulate those high catches that come to natural host odor. At the same time, work going on by people like Chris Green, looking at how do tsetse flies use visual responses? So going from left to right, we know that in case of morsitans, a warthog is something like 80% of all blood meals come from warthog. What is it about a warthog visually that also contributes to it? So we have a real warthog, a stuffed warthog, a warthog model, and we find that if you change the color slightly, you make it bigger, change the shape slightly, you can end up with a super warthog. The upshot of all of that, again, something like 20 years work, were these large targets which simulate natural hosts. They're about, so one's here, just to give you an idea of the thing. So we put out these 4 per square kilometer. They're baited with the odors, which you can see on the picture on the slide there. They're in sachets that holds the octanol, acetone, phenols. It's sprayed with insecticide. And Tsetse are attracted to it. And again, in that low percentage, we only have to kill something like more than 4%. And we can do that with this sparse deployment of targets. About the time I was involved in some of that work, and we were all excited and very pleased with ourselves. And then an MSc student came along and said, well, why don't we use cattle? They're in these sorts of areas. What happens if we just treat them? We don't have to get natural artificial odors. We don't have to build a target. And she showed that indeed these cattle were, if you treat them with pyrethroids, susceptible to tsetse flies. Tsetse flies were susceptible to pyrethroids. A bit more work on that, and if we can even make this slightly more elegant. It turns out most tsetse flies feed on the legs and belly of cattle, and we can just treat those areas, and without using too much insecticide, we should be able to kill tsetse. So what were the impacts of those two? So again, this very briefly, this is what happened in the 80s. Aerial spraying dominated, with ground spraying in the early part of the 80s. Targets were developed in the mid-80s. Cattle started changing from an organophosphate to a deltamethrin, and that just overtook in Zimbabwe as a way of controlling tsetse flies. With that change, cases of animal trips went down from something like 10,000 cases a year down to single figures. And you can see it's not because we weren't looking, they were still surveying intensely. The insecticide-treated cattle and the sort of restricted application technique, that was adopted in Uganda and became part of the Stamping Out Sleeping Sickness campaign. And again, you can see here, this is the global figure that I showed you about. And again, just like DRC is for Gambian hat, Uganda is the big center for Rhodesian hat. And you can see as the numbers of cases go down in Uganda, the numbers globally go down. And treating cattle was a big part of that effort. But the big problem is Gambian sleeping sickness. And it turns out that all those technologies which were highly effective in Uganda and in Zimbabwe and in other places in East and Southern Africa were just not good for the vectors, the riverine vectors of Gambian sleeping sickness. The attractants, oxynol, acetone, and so on, were not effective. These flies didn't seem to be affected by it. These big targets were very cumbersome if we had to put out lots of them. In many of the places, there aren't many cattle. And there was some efforts at using traps to control tsetse, but these were cumbersome and expensive. So basically, vector control, as I said, was just not a part of controlling Gambian sleeping sickness. So about 15 years ago, we at school and other partners in Greenwich, in Kenya, Uganda, and across Africa, started a program of looking at, can we get something better for riverine tsetse? So we looked at their odors, we looked at their visual cues, we revisited all of these things using many of the technologies developed in Zimbabwe and East Africa. So first of all, are the odors different? We tested things like acetone and octanol. They don't seem to work, but is there something else in cow odor, for instance, that's important? So again, this catch index, which I showed you earlier, we have highly responsive sort of 8-fold increases with morsitans and pallidipes, morsitans, savanna flies. Basically, all these riverine species at the top here, Palpalis, gambiensis, and so forth, either unresponsive or only slightly responsive. So that was with ox odor. Perhaps we're using the wrong odor. Perhaps there's another odor that we should be using. We know what these feed on. In particular, monitor lizard, important vector host, host for tsetse. Again, that doesn't seem to be very important. It just seems that these flies are just not highly responsive to odors. At the same time that we were trying to look for this elusive odor, which turned out didn't exist, Jenny Lind was a newly appointed postdoc here, come from the world of mosquito biology, and she decided she would look at visual responses and used electric nets to look at how many flies come to different colored and shaped objects. So revisited the whole visual response of tsetse, but with these particular flies. So one experiment she did was compare the numbers coming to different size objects. In the past, we had always thought a big object, hence the big targets, attract more. That is true, But if we look at this, start to think of how many flies per square meter, so per dollar spent on material, for instance, what we find is that as you get smaller, the flies caught per dollar per area of cloth actually increases. It's 9 times better to use that small target than have a big target. Jen also looked at different colors, again, using these small electric nets. So a whole series of different colors were looked at. And the upshot of all of that work, again, this covers something like 5 or 6 years of work by Jenny and others. We see that all the species, the riverine Tsetse, are highly responsive to these very small targets. Morsitans aren't. And we also find this general trend of yellows, greens, whites, less attractive as you go to this blue color, highly attractive. So the upshot is you need something small, blue, and you end up with a tiny target. And so rather than that big thing that I showed you earlier, we have this thing. And Tsetse are attracted to the blue object, fly around it, collide with this netting, which is also impregnated with insecticide, pick up a lethal dose of insecticide. So that turns out to be twice as effective as the devices that were used, those traps, 10 times cheaper, longer lasting, easier to deploy. You can put it in your backpack, put it in a pirogue, go down a river and deploy them along the riverbank. So we started doing trials on this. And what did we find? So this, first of all, is Uganda. Again, I've framed these things rather than giving the catches, just as catch indices, where by definition, at the beginning, before we put out targets, catch index is 1. So a decline would show, as we see here, goes down from 1 in the very first couple of months and goes down very rapidly. Within about 6 months, is less than 20% and stays at about 5–10%. In a neighboring area where we didn't put out any tiny targets, the inset there, again, you can see, yes, the population varies, but it never consistently stays below 1. So deploying these tiny targets at once every 200 meters along these rivers seems to have driven down the population. And then this is in the Mandul focus in southern Chad. Again, the same sort of thing, putting out traps. Again, by definition, it's 1 at the beginning, and then it looks like, well, we look some months later, and it looks like a pretty rubbish monitoring campaign, but actually what was happening, every 3 months we were going out looking for Tsetse flies and actually catching nothing. So in this particular setting, it seemed to have even eliminated flies. So those early trials then led to a massive expansion in the technology, and it was adopted by many countries. So this is the figure at its peak. So sort of headline figures, 21,000 kilometers covered with this technology of tiny targets. That area is something like 2.5 million people. And those countries that listed here, those represent 90% of all the cases. So this technology has gone in the space of 10 years, really rolled out in the countries where it's needed most. So we can show impact on Tsetse flies. What about impact on disease? So I think one of the best examples is the Mandul focus. So that's the place where we saw those catches collapse. And the vertical bars there are when we are putting out target. The bars are targets, and the catches you can see going down. The dashes are where we look and catch nothing. So in this particular area, numbers of Tsetse go down by 99% in our monitoring traps. And we reckon, so here, this is work by Cat Rock. What she's showing is the numbers of new infections before the intervention. So that's the gray plots. And then afterwards, what her model would have predicted would have happened if we hadn't introduced vector control. So it would have shown this decline, but still would have been hundreds of cases. The consequence of adding vector control was that the cases went down to low tens. Numbers of new infections were order 5 or 6 per year. And subsequent analyses, we basically have interrupted transmission from 2016 onwards through the deployment of those targets. So these are the national figures for Chad, going down from hundreds of cases down to this, in this decade, 2023, I think is the most recent, down to sort of a few 10 cases or so, tens. And we see similar sorts of trajectories in all the countries where we've been deploying targets. So Chad, Uganda, Cote d'Ivoire. All of these countries announced elimination of sleeping sickness as a public health problem. Tiny targets have now become part of their national policies. It's adopted as part of WHO policy. And more countries have adopted it and are likely to follow these declarations. So going back to that earlier figure about those elimination goals. So that was the 2020 goal was 2,000 cases. Actually, that was achieved a few years earlier. And, you know, does it look as though we are on our way to achieving that elimination goal through these efforts? And in some ways, we might even do better than that because some of the other things are going nothing to do with entomology or clinicians, but just global change. So we're looking in East Africa, we've been looking at the edges of parks, which are sleeping sickness foci for Rhodesian hat. And so that's Serengeti, Simanjiro, which is a district on the edge of Tarangire. And we've been looking at Tsetse in those particular areas at the interface between conservation and wilderness areas, farming areas. Basically, what we find is that as you go out of the parks, the numbers of Tsetse are very low. So minus 5,000, that's 5,000 meters into the park, 5 kilometers in, and 5,000 is out. So as you can see, by about 5 kilometers from the park edge, we don't find any Tsetse flies. So Jen Lord led this work, used some very nice modeling and using remote sense data. And is that the button? Which is the button for? Red one? Is that working? Anyway, the blue areas are basically where we're not predicted not to find Tsetse flies. And you can see it sort of follows the edge of the park. So the red line are the boundaries of the park. So north of that, you can see this big blue patch. And basically what's happening is that settlement, farming, people's activities are degrading the habitat that's particularly important for savanna flies, basically making it uninhabitable for Tsetse. So that just anthropogenic change of farming is actually controlling Tsetse outside the park. We can see to the left of that map, to the west, we can see this patch. Now that does look as though it should be good for Tsetse. But actually, when we go to that area, we can't find Tsetse in that particular area. Even though the habitat, there's a picture of it, that looks great for Tsetse. But what we also did at the same time was speaking to livestock keepers in that area. And it turns out they were using their own resources without real knowledge of the government, were treating their cattle with pyrethroids and were basically doing Tsetse control led by farmers. And looking at the figures that we can see, the sort of the amount of insecticide used and how it's applied, putting that into our epidemiological models suggests that basically farmers themselves are controlling sleeping sickness and the spread of sleeping sickness out of the park. So farmers and humans themselves, just in our general activities, are perhaps having a big impact on Tsetse populations. Going further south in Zimbabwe, what about in the park there? This is the Mana Pools National Park, been a park since the 1960s. There's been no change there, lots of Tsetse flies, lots of hosts. How are Tsetse fly populations doing in this particular area? So when it became a research station, it was also set up as a Met station. So we actually have continuous Met data for this Tsetse research station from 1959 onwards. And I'm just showing you here what has happened. So these are from just showing it from 1960 through to 2017 when this paper was published by Jen Lord. And on the face of it, on that top figure, it doesn't really look that striking that much is changing. You see, yeah, there's a slight drift up. You can see the peaks. There's more of those above 30. But if we look at the general drift in the figure on the bottom there, you can see that in that period, it's gone up about 1 degree Celsius average temperature, as a consequence of climate change. Looking at a bit more detail, though, what we do find, look at how each month has changed, again, over that same period. And if we look at October, November, December, those have gone up between 1 to 2 degrees. That is the hot season in Southern Africa. These are temperatures in Zimbabwe this time of year. You would typically reach a very hot day would be in the low 40s. And those days are becoming more frequent, and the average is higher. So we can put those temperatures, put that in models, and we have some very good relationships, again, because these flies lend themselves to this sort of study. And the top ones look at mortality rates. Look at the x-axis. So as you get above 30, the pupal mortality shoots up. So if it gets into the 30s, pupal mortality is going to be very difficult for tsetse flies. And you look on the left there, what are the mortality rates? Again, we only need 2%, 3%. You can see these are in the ranges that are very difficult for tsetse fly populations. So what Jen did was took the temperatures from Recometry, plugged that into a model, and then predicts what would happen to a population. And to check that, she also used catches from cattle. And we've been doing this routinely, again, since the 60s. Send out a team, they collect tsetse flies, they're fed, brought back to an insectary, and we test insecticides. So it's a routine thing that had been done for a long period. And she puts those together. So in the past, so in this figure here, in the in the 1980s, we could catch loads of flies. So it wasn't really, we couldn't really work out whether the numbers were going down. But from about 1990 onwards, we began to get a little bit difficulty getting enough flies for insecticide testing. And those are the red dots that you can see here. This is the numbers of tsetse that would be captured on an ox. And you can see the numbers collapsing, and Jen fits the model to that. So it looks as though the trend that we are seeing in the numbers of flies at Recometry in Zimbabwe, in an area where loads of hosts, habitat hasn't changed, is also declining. So those are the pressures on tsetse populations, and also drugs are getting better. So again, in the last few years, there's been now a new oral drug, very easy to administer. In prospect, 2025, we'd like to have a drug, single dose, will treat the disease. So this all sort of adds to the sense that perhaps we have solved the problem. Have we? Are we doing? Okay. So have we solved the problem? It seems certain. It looks like the figure on the left there, Kilimanjaro, snows are going. It looks like we're going to put a lot of pressure and the tsetse flies will go with it. But I think that's not quite the case. First of all, there's going to be shocks. So this gives you an example. I showed you those three figures of those countries. In fact, the fourth country also had tiny targets at the beginning, was Guinea. It had an Ebola outbreak that interrupted all interventions, and other countries will face those interruptions. So that sort of steady progression, elimination, is that going to happen? Possibly not. The other thing that we're doing at the moment is we're scaling back tsetse control, where this is the case of Uganda, work by Josh Longbottom. The figure on the left shows where we put tiny targets, 4,000 square kilometers deployed as we expanded over that area up to the end of the last, up to 2019. Since 2023, we haven't deployed any targets. There's no tsetse control going on in that area. Will we go back to that pre-intervention setting as we have? So what we would predict is that if we stop tsetse control, tsetse populations should recover very rapidly, something like 1 to 2 orders of magnitude per year. We should get back to that pre-intervention levels within 3 years. This is the case of Maracha, where we stopped control in that district in 2020, and there's been no control since 2023. We do see some recovery. These are the red dots here that you can see, but really it's going nothing like, or plotted on a logarithmic scale, nothing like we would predict. So for some reason, which we don't understand, tsetse populations are not recovering as fast as we would expect, but they are not disappearing. What about Zimbabwe? So the models that Jen Lord produced, Josh then again took met data from all met stations and then could see the relationship between remote sensors of temperature and measures. So he could then apply her model across northern Zimbabwe and predict what would happen to populations across that region. These are the outputs. It's not very clear in this particular figure. What he's doing is contrasting 2001 to 2005 to 2012 to 2016. But conveniently, he has another figure. So basically, blue areas are where tsetse populations have gone down, red areas where they increase. What he predicts is that as the world gets warmer, basically the tsetse populations will move into higher elevations. So with them, the foci could also change. So we might not be getting rid of foci through climate change. We might just be moving them around. What about this collapse that Jen Lord, has that happened? It looks like in 2017, we were about to eliminate tsetse flies from the Zambezi Valley. Did that happen? So conveniently, Glinvale and others were repeating an experiment that Leggett and Pilson did in 1959-60. We could go back to the very same place, do the same method with this Vatox, and see how many flies we caught and compare our results with theirs. He did that in this period. This leads up to the moment when Jen Lord published her paper, and for Polydipes, which is what Leggett and Pilson were catching, we can see this very acute decline. Sort of fits with what she reported. But what happened in the last few years is that numbers have actually come back up again. They're back to the levels that they were in 2012. So what happened there? So this is with all the data. So, and this is the mean that Jen uses in her paper. So the 2016, which is the last year of her analysis, it was indeed a very, very hot year. Look at those temperatures above, especially in October, November. But the very next year, actually, the numbers were back down to sort of normal levels. And actually, that November, that crucial period for tsetse flies, was actually quite cool. So I think what's happening is their response to year changes. But this is not to say this is not climate denial or anything like that. The numbers in Zimbabwe are only a fraction, about 10% of what they were in the 1960s. And lastly, just want to talk about other places like, and in particular, the zoonotic form of the disease, so Gambian Rhodesian hat. So this was the numbers going down. I showed you this figure of Uganda, and then there's this uptick. So what happened there? Uganda was doing well. Uganda seemed to be the main place where we should be focusing, but in the late teens, suddenly there's a surge in cases. And that was all to do with Malawi. And in fact, it was all to do with one small place in Malawi. Bwaza Marsh suddenly had an outbreak of sleeping sickness. So that's work that we're currently doing with Chris, Karina Mondragon and Shem, and our partners in Malawi. And the cases are all outside the park. And so what our hypothesis was that flies were coming out of the park biting people. So Karina and our partners in Malawi have been conducting transects along that park. And what we find, just like in Tanzania, the numbers, we don't catch any tsetse flies outside the park. So somehow humans and flies are still meeting, but we don't really know where that contact is happening or how to deal with it. So is the problem solved? I think we're making obviously great progress. Lots of countries are approaching elimination. We've got some great methods of control, got some good things coming in prospect. But we also have future challenges. So we haven't solved the problem, we've just got new problems. I'm going to skip. I was going to suggest some solutions, but some interesting things to do, but I'm not going to do that because I think the last important thing I wanted to say was this. That all the advances have come about through early career researchers conducting largely discovery research. So tiny targets, we were all looking for odors. Jenny Lind started to look at visual responses, and she discovered tiny targets, and that has changed the game, as you can see. And as I was writing this, I was reflecting, actually, that has happened consistently. So Mary Owaga, when everyone said it's in the smell, the breath of animals, she looked at other products from animals and found what basically were the phenols. Madeline Thompson, looking at pyrethroids on cattle. Chris Green, looking at color. They were all of those were either MSc students, first appointment as MSc students, as postdocs. Glinvale's work, he was a PhD student doing it. So it's to sort of say the answers and the future is in the hands of young, inventive scientists who don't come to it with preconceived notions and are not the tired people that Chris and I are. There's loads of people, sorry I've gone on a bit too long there. There's loads of people I'd like to thank I've just listed some names here because I sometimes bump into them in the corridor. Thank you.
Perfectly fine. Thanks, Steve, for taking us on that journey. And we've got about 8 minutes for questions. So I think we'll start here. Myron.
Hi. What do you know about the insect trypanosome interaction? Does it have any impact on the insect hosting it? Does it always host it?
Yeah, so there is a lot of work on that. So our colleague Alvaro, who is at the school, is now in the US. That is his big area of research. So the first bit when you asked that question, I was thinking the answer is actually Tsetse are very refractory, are not very good vectors. They are largely refractory. So in the case of brucei, they can only probably be infected in their very first blood meal. And after that, they can't be infected. So there's a whole lot of, that interaction about their ability to be infected or withstand infection. And then does it have any impact on their feeding behavior or sort of are infected flies? So there is some evidence that they probe more. For instance, there's not a lot of work on looking at that, but there is some evidence. I think the trouble is it's quite a difficult thing to especially test in the field. The infection rates are less than 1 in 1,000 for brucei. So really difficult to do that. Yes, you could do experiments in a lab with infected flies, but making that translation to the field is difficult.
Ben, and then we'll go to Richard.
Thank you. So why do they last 6 months and not less, not more? How can you make them last more than 6 months?
Targets.
The tiny targets, yes.
So the limit to it is in fact the insecticide. So this is impregnated with deltamethrin. It's the same sort of technology that's used on bed nets, but of course it's outside and it's rained. So yeah, if we could get it to last longer, that would be a good thing. So you can sometimes to do with the loading, so the amount of insecticides you put on there. At the moment, we haven't done that. In some places, we do also lose quite a lot through flooding, because you have to put them along rivers. So whether that is a limiting factor, but yeah.
Okay, we should pass to Richard and then I'll.
Thank you, Steve. That really is properly important entomology, and it's super to highlight the role that students and early career people are playing too. My question is whether there is as yet any sign of resistance to pyrethroids in Tsetse.
Yeah, so no, there's not. And I think just to give you some background, we are just using, in all the methods that we use, we're just using deltamethrin. Insecticide-treated cattle, aerial spraying, ground spraying, targets. So Tsetse populations are that's all they're exposed to. And at the moment, no insecticide resistance has emerged. So why might that be? We think possibly the low reproductive rate of the population, the small size of the population. In some cases in the past, the interventions tended to be not continuous, but they are these days. These targets are put out for long periods. So I wouldn't say it will never emerge, but at the moment, it doesn't seem to be a problem that faces us.
So I just wanted to know the impact on the food webs and the ecosystem as a whole. So was there a bycatch to the small traps, and also was there an impact on the predators of the Tsetse flies?
So quite a lot of this sort of analysis of behavior, it turns out not many insects are doing this sort of thing. So for instance, almost like the color is the flip side of plant feeding insects. We have yellow, don't we? Have yellow traps for aphids, and this is the opposite of that. But there are, of course, things like tabanids might be attracted to Stomoxis. So Some of those insects are raw, but again, they don't have the sort of population biology of Tsetse. They don't have this very slow reproductive rate. We're not actually killing, you know, it's only like probably 4% of the population. So we don't seem to have any impact on other populations, and even quite gross interventions like in the Okavango, where they have eliminated tsetse flies completely. There is not a tsetse flies in the Okavango anymore. Didn't seem to have any other insect disappear, even though they looked for that impact.
It's time for two more questions. So that's the lady up. Okay.
Thank you. I recall there was a program of releasing sterile female tsetse flies into the population to try and control them. Do you have any thoughts on that? Do you remember that in Zimbabwe?
Yeah, so releasing sterile, in fact, sterile males, yeah, to so yeah, there's been used, for instance, on Zanzibar, and there's interest in that. There's a personal, I should declare a conflict of interest here, because I published work arguing why we think that is a very, very bad idea. It's partly because we have some very good insecticidal techniques which work very quickly and effectively. Whereas sterile insect technique, because of the sort of reproductive cycle of Tsetse, and they are long-lived, even if you're sterilizing them, it takes a long time to have an impact on that population. Secondly, males and females are both vectors. So if you introduce sterile mosquitoes, you don't add to transmission. If you introduce sterile male tsetse flies, you are adding vectors. So yeah, and there's a lot more to that. But anyway, basically, yes, it does exist, has been tried. I don't think it's a good idea.
Okay, yeah. And then we'll finish with Chris.
Thank you. That was a great talk, actually. My question is is there any biocontrol methods to use? Because using pesticide will harm the untargeted insects. So thank you.
Yeah, again, that's the sort of area that before I start, so in the 70s, really, was interest in biocontrol agents for Tsetse. So they do have parasitoids and to predate the pupae in the ground. Robber flies, bendix wasps attack them while they're feeding. But the sort of mortality rates are not sufficient. We can't breed up these sorts of things in large numbers. The pupal stages are often, apart from in the dry season when they are concentrated, widely dispersed. So it doesn't look as though you can get one culture those things to act as biocontrol agents. And I suppose it's also part of, again, that sort of biology that these flies do not seem to be, they are sort of in sort of older population ecology language of the K-selecting, very slow growing, low mortality rate, look after their young, and we can't really manipulate them in those sorts of ways.
Thanks for an excellent talk, Steve. You might have just answered the question I was going to ask, but I was going to ask about whether there are any natural enemies of tsetse fly and Hymenopterus parasitoids, but you said there are, so yeah, and predators as well, presumably.
Yeah, so lizards, birds. So there's an interesting there's some interesting things again, because they're such big flies, you can watch them and see. So for instance, you can see flies feeding on a cow, and if a bendix wasp arrives, they all disappear, which is a predatory wasp. And asilid, so robber flies, you can see them attacking tsetse flies and taking them.
Okay, coffee. So just one last round of applause for Steve for a wonderful talk this morning. And thank you for all your questions and making my job a bit easier. Steve will be around to answer any questions in the coffee break on tsetse, fishing or football.